Let's talk some more about signal transduction. So we're going to look at the first step of signal transduction in the screencast, which is reception of the signal. So reception is the first step in a signal transduction pathway. And this happens when a signaling molecule binds to a receptor and it causes it to change shape, okay? And of course, if we're changing the structure, we're going to change the function. So that's kind of, structure and function is like hinges on this whole entire pathway, signal transduction pathway, okay? So first happens is some kind of a signal molecule, which we call a ligand. It's another name for a signal molecule. The bond between a ligand and a receptor is very, very specific. So one ligand bonds to a specific receptor, and that receptor can only recognize a particular ligand. Okay? Once the ligand binds, it induces a change in shape in the receptor. And that helps to transmit the signal across the plasma membrane. Okay? Most receptors are plasma membrane proteins. So let's look at some of those. Okay? Any molecule that is water soluble, i.e. polar and hydrophilic, bind to receptor sites on receptor proteins that span the plasma membrane, so transmembrane proteins, and they bind on the extracellular side. There are three main types of membrane receptors that we're going to look at. One is a G-protein coupled receptor. Second one is a receptor tyrosine kinase. And the third one is an ion channel receptor. So let's start with G-protein coupled receptors. So G-protein coupled receptors are the most popular of cell surface receptors and the most plentiful. So it is a plasma membrane receptor that works with the help of something called a G protein. And the G stands for GTP, which looks similar to ATP because it is similar to ATP. Okay. This G protein acts as an on-off switch. And if GDP is bound to the G protein is inactive. If GTP is bound to the G protein, then it is active. Okay, and let's take a look and see how that works. So here's what an example of what a G protein coupled receptor would look like. Okay, you see it's a transmembrane protein. It spans the entire membrane. There is an area outside the cell where the ligand will bind. Binding site of the ligand, and there's an area inside the cell where the G protein will bind. Okay, and this is just kind of a little cartoon of how it happens. So here's my G protein receptor, here's my G protein, and here's some kind of an enzyme. Okay, when our signaling molecule or a ligand binds to the G protein coupled receptor, okay, it causes GDP to be released from the G protein and GTP to bind. This activates the G protein. And then let's go down here to step 3. Once it's activated, the G protein goes and binds to the enzyme and activates the enzyme. And then the enzyme releases something that can cause a cellular response or response in the cell. Okay, and that something is almost always a phosphate. Okay, so again, when the ligand binds up here, it causes a change in shape in the receptor. That'll cause a change in the G protein. The G protein goes over and activates the enzyme. Change in shape of the enzyme, and the enzyme triggers a cellular response. Okay, so that's our first kind of receptor. The second, numero dos, is called a receptor tyrosine kinase. Okay, and a kinase, you see that ASE, which means it's an enzyme. A kinase is an enzyme that adds a phosphate group to something, to whatever it's supposed to, okay? So, and receptor tyrosine kinases are receptors that attach phosphates to the amino acid tyrosine, okay? And not all kinases do this. Kinase is like a general term for any enzyme that attaches a phosphate group to something. A receptor tyrosine kinase is a specific name for a receptor that attaches phosphates to tyrosines, okay? And the cool thing about receptor tyrosine kinase is that it can trigger multiple signal transduction pathways at once. So whereas Gprotein coupled receptors can just trigger one signal transduction pathway, receptor tyrosine kinases can trigger multiple pathways. Okay, when these don't work, when our receptor tyrosine kinases don't work or work actually too good, it's associated with cancer. So here is a receptor tyrosine kinase. Okay, and when it's inactive, okay, it's in kind of two pieces, one monomer. So one here and one here. Okay, then we have our ligand come in and binds to the receptor in the binding site. This causes these two to come together, okay, and dimerize to become a dimer. So we're getting some quaternary structure here, okay? Once that happens, once these two come together with the ligand attached, the kinase, the receptor tyrosine kinase becomes activated. And it begins to phosphorylate stuff like crazy, but the first thing it adds a phosphate group to is itself, okay? Once it adds phosphate groups to itself, then it becomes fully activated and it can begin to phosphorylate other things. So what it does is it phosphorylates these relay proteins, adds phosphate group to them, and then they go off and generate some kind of cellular response. And because there's so many binding sites, it can generate more than one cellular response at a time. Okay. So that's how receptor tyrosine kinases work. The third type of receptor we're going to talk about is a ligand-gated ion channel. Okay, and a ligand ion channel receptor acts like a gate when the receptor changes shape. Okay, so when a signaling molecule or a ligand binds to the receptor, the gate opens to allow specific ions like sodium or calcium through a channel in the receptor. And sodium ions are associated with nerve impulse conduction. And calcium ions are associated with muscle contraction. MUSCLE. Okay, so let's see how these guys work. So here is our ligand-gated ion channel receptor. Its normal position, the gate is closed. When our ligand binds, this causes the gate to open. Once it opens, it's a channel protein. And basically, if you fit through, you can go through, and it's usually a very, very small channel so that something like a sodium ion can truck on through. Okay. Once and then the influx of ions will trigger some kind of cellular response. And when we get to the response portion, we'll talk about what those responses can be. Once the response happens, the ligand is released and the gate closes back to its normal position. Okay. So those are the three types of transmembrane receptors. There are also receptors that are inside the cell, intracellular receptors. Okay, so intracellular receptors are found in the cytosol or the nucleus of the target cells. And these work well if the ligand is small or hydrophobic. Okay, these guys can cross the membrane itself. Okay, and the whole point of receiving a signal is to get that signal into the cell. And so the receptors kind of translate it across the membrane. But if you have a small or hydrophobic ligand, they can cross the membrane themselves and get their message into the cell. Okay. Examples of hydrophobic messengers are steroid hormones and thyroid hormones in animals. And so the hormone will get into the cell, find its receptor inside the cell, and then become a hormone receptor complex. And this can act to trigger a intracellular response, a cellular response. A lot of times these act as transcription factors, which are used to turn on genes. So let's see how this works. So here we have a hormone. Here's some testosterone. It's a steroid hormone. It can go right across that plasma membrane because it's small and hydrophobic. Here's its receptor. Once the hormone is inside, they get together and find each other. Now I have a hormone receptor complex, and this can act inside the cell to trigger a cellular response. Most of the time, these guys act as transcription factors. They go into the nucleus, bind to DNA, and activate transcription, which makes a messenger RNA, and then that messenger RNA will make a new protein. Okay, so that was reception. And next, we're going to talk about transduction. So head on out to the next screencast.